Hey y'all, welcome back again to another episode of Healthy Design. I thought today we'd go back and answer the question from Sarah dealing with methylation and supplements, MTHFR. We're probably not actually going to get to the answer for that question today because I feel like we need to do a little background first to make the answer to the question make sense. So we'll give a little background today on genetics and epigenetics, and then also the methylation pathway. And then next week we'll kind of move into MTHFR specifically. So genetics is the study of heredity. Kind of focuses on genes, variations in the genes, and then how those traits are passed on. And we can have different, genetic disorders where those disorders are related to abnormalities in those genes. So it's an inherited medical condition caused by a DNA abnormality. Some examples of this include Down syndrome, Huntington's disease, and also cystic fibrosis. However, most disease processes that we have and see are the more kind of common ones, or are going to have a genetic component, but then there's also a big epigenetic component. And so epigenetic is the study of how environmental factors and behavior influences gene activity and how it can alter a DNA sequence. And so like with high blood pressure, diabetes, if you have that, most likely you have some genetic SNPs, some alterations that make you more susceptible to it, but then you also have to be exposed to certain things for that to be expressed. So, it's possible that you could have that susceptibility based on the things that you're exposed to, though those genes never get turned on and you never express diabetes or high blood pressure. Or vice versa, you could have that susceptibility, be exposed to things that turn those genes on, and then that does get expressed and you do have that issue. So also with our genes, I can't find an exact number. I don't, they're still researching and doing things, and so they still don't know. The best I can find is that there's 19,000 to 20,000 protein coding genes in in the human genome, but there's also thousands of non-protein coding genes, and there's not an exact answer on that. And it should also be noted that when they started to try to map the human genome, they thought it was going to answer everything, thought they were going to be able to cure everything, and it just hasn't happened. It's a whole lot more complex. There's a lot of redundancies in the body, which is a good thing. That way, if one area is not working, there's all these others that can kind of come in and kind of fill that gap. So if there weren't these redundancies, we probably wouldn't have survived as long as we have. So that is a good thing, but it can make it hard to kind of figure everything out. And so with what we're talking about today, we're not going to be talking about true genetic disorders like Down syndrome or anything where there's a true gene abnormality or chromosomal abnormality. What we're talking about are these SNPs. SNP, that stands for single nucleotide polymorphism. And so those SNPs, so it's a change in just that single area, but that's going to change the structure of that protein that gene's coding for. And as we talked about before, structure determines function. So once you change that structure, now you've changed that function. Depending on where that change occurs is going to determine what change in function there is too. So you can kind of see how this could get very complicated very quickly. And so it does appear that with some of these SNPs, it can actually make that gene run faster, and with others, it can make it run slower. Either way, what we do know is it makes the function of that gene, it makes it less efficient. And so a lot of times these are coding for different proteins that are involved in, or they're enzymes to help speed along different reactions or make different reactions occur. And that's exactly what MTHFR is. So the good thing with this is we kind of see that, the genetics themselves don't give us the whole picture. And we have no control over our genetics. And so with more these common diseases, quote unquote common diseases, there is a huge epigenetic factor, and that we do have some control over. So that should hopefully be a positive message, something that's kind of encouraging. we still don't have ultimate control because there still is that genetic component there that we don't have any control over whatsoever. But we can we can at least control some of the exposures we have. We can control what we eat, how much sleep we're getting, different things like that. And the reason why I kind of keep saying common diseases, a lot of the true genetic disorders or inborn errors of metabolism, the outright big issues, those tend to be pretty rare. Again, there probably could be some minor or more minor changes going along that could still lead to some different effects from a functional perspective, but not, most of those do tend to be like, if they're really bad, they tend to be very life-threatening and short lifespans. So that's why we're not going to concern ourselves too much with those today. Not that they're not important by any means, but it's just kind of beyond what we're talking about. So let's move into methylation, because this is where MTHFR plays a big role. Again, it's not the only gene, though, involved in methylation, so that's something to keep in mind, too. Because I do think the way it's portrayed, most time people think that's the only one. And so if it's got an issue, well, then they have this big issue, and then they're stuck with this. And that's just not the case. Again, there's all kind of redundancies built into the body, and so it's pretty smart. So sometimes we just got to keep that in mind. But methylation is a biochemical process that involves the transfer of a methyl group, which is CH3, so one carbon atom and then 3 hydrogen atoms. It's involved in regulating genes, synthesizing DNA, producing certain neurotransmitters, dopamine, serotonin. It's involved in detoxification and also in kind of metabolism of estrogen, and it's also involved in immune function. So it's involved in a lot of different things that can have a huge impact on our health. So when genes are methylated, normally that ends up turning the gene off, so it's not expressed. So again, if we have different genes we don't want to express, like different ones that can make us susceptible to cancer, to diabetes, we want those to be methylated and turned off and not expressed. That's a good thing. So we want this pathway to be working well. So let's move on to the pathway itself. So this is a little biochemistry for you. Again, we're going to try to keep it as simple as we can and just focus on what's important. So we've got different things. So we eat food, and if we eat protein, we get different amino acids in, one of those being methionine. Methionine can kind of be broken down and get to homocysteine. And so what can happen then, though, is that homocysteine, which you can do a regular blood test and get that number. Most often people are high, which does show there could be an issue with methylation. There are other things that could lead to that being elevated too. So it's not necessarily just a marker of methylation. But when you have that homocysteine, it can be methylated back into methionine. And there's 2 pathways it can go. It can go through the folate pathway, which is where MTHFR is going to come into play. So it's a folate dependent pathway. So we do need methylfolate to be able to get that homocysteine back to methionine. There's another pathway that I've heard it pronounced different ways, betaine or beta-ene dependent pathway. Another name for beta-ene or betaine is TMG, trimethylglycine. So let's maybe go with that one, because that way you see the methyl group, or you hear methyl in it, kind of helps you out too. So that is another pathway that can help get that homocysteine back up to methionine. However, that's not the primary way to do it. It can be used in cases of emergency, is kind of a way to think about it. So we want it to function well, but we don't want just it to function well and not that folate pathway. We want it to function well too. We want them both to function well. And so with the folate dependent pathway, we're dependent on certain nutrients as cofactors for some of those chemical reactions to take place. Folate's obviously one of them. Vitamin B2, so riboflavin, vitamin B6, pyridoxine, and then B12. So, and we could do like a methylcobalamin for that to help have that methyl group to donate. The TMG dependent pathway is dependent on glycine, so TMG. It's also dependent on zinc and choline. And choline's another amino acid you can find a lot in eggs and different things too. I do want to note there's other enzymes involved in methylation, MTR, MTRR, MTHR, and then BHMT. And then there's some other pathways we'll probably get into at another point where CBS is involved as well. Not the news or the TV station, but it's a gene. So another key methyl donor that we haven't mentioned is S-adenosyl methionine. So it's a kind of universal methyl donor, and it will donate its methyl group for 200 or more different functions in the body that require it. And so once it does all that, it gets broken down into homocysteine, and then we want it, kind of going back to the methionine that we talked about. The CBS enzyme that I mentioned, this is where it comes into play. Homocysteine, rather than going back up to methionine, it can actually go down through another pathway to produce glutathione. Glutathione is our master antioxidant, so if we're under a lot of oxidative stress, then that homocysteine, rather than going back up to methionine, might actually start going down this pathway to produce the glutathione to help deal with that oxidative stress. And so for that one, we obviously need that CBS enzyme to work well, but one of the cofactors for it is vitamin B6. So this is why you see a lot of times people will want to supplement a lot with the B vitamins for some of these things, with MTHFR and other stuff, because they are cofactors for so many different reactions that are occurring in the body. And so another thing with that CBS enzyme and stuff, these can end up going down another pathway into the Krebs cycle, which is used to produce energy. And as we kind of saw with the betaine, beta-ene, TMG, you got all these different names for things. Well, for the Krebs cycle, it's also called the carboxylic acid cycle, tricyclic acid cycle. So I mean, there's all these different names for the same thing. And so that can make some of this confusing to know which one they're talking about, because it sounds like people are talking about something else. And so for energy production, some of the vitamins and different cofactors we need: vitamin B1, vitamin B2, vitamin B3, vitamin B5, vitamin B7, vitamin B12. So again, you see where the B vitamins come and play a role in energy production too. And so that's why when people are low on energy, they talk about that, talk about iron as well. So magnesium is another one to keep in mind. Basically, whenever there's ATP, there's magnesium. So ATP is the energy part, but magnesium is also involved with SAMe, glutathione, and the folate cycle too. So we want to make sure we have adequate magnesium, and that is something that a lot of people are deficient in. People will vary on how much they're deficient in different B vitamins. Some may not be at all. And then sometimes too, we got to keep in mind as well that maybe it's not even a diet thing. Maybe they're getting sufficient amounts via diet, etc., but because of stress or that oxidative stress, different things, that's kind of depleting some of these nutrients rather than not getting enough. And so that's why we want to look at the foundations of health and not just focus on, oh, my B6 is low, so I just need more. Maybe it's not that it's really low, maybe it's just you're so stressed that it's getting used up. And so that way we can manage stress and fix things rather than having to take a pill of any sort. So, all right. With the energy cycle too, one thing I wanted to mention, there's two different points where different amino acids can come into that cycle. So, think of the methylation cycle path, the folate pathway, the betaine, TMG pathway. They're kind of like these circles that just kind of keep running as needed. And so the same with our Krebs cycle to produce energy, it just kind of keeps running. So there's different points in that where different things can come in. And what you're trying to do is get glucose in there to produce energy, produce ATP. And so this is why if you're eating carbohydrates, because you'll hear people say all the time that carbohydrates are protein sparing. This is how carbohydrates are protein sparing. If you're eating enough carbohydrates, then you don't need those amino acids to come in at those different points to keep that cycle going. And so that way it helps to spare protein instead of using it for that. Let's see. So again, I think that's really all we'll say regarding the methylation pathway. It's definitely a simplified explanation for it. I hope it kind of helps to get across like how complex it really is and just how complex our body is. And then also again, how there's all these different redundancies, like, the homocysteine can go back up to methionine, but if it needs to, it can go to glutathione, but also if it needs to, it can lead to different things to produce energy. So, it's just kind of depends on what the body needs. So it's got all these different ways around it. And then again, if one thing's not working, there's something else that can probably come in and help out. And so, some compounds in our body have to be methylated so they can be used by our body. So that's why, this methylation process is very important. And then there are some compounds that need to be methylated to be expelled by our body. And so this might be interesting to people. So some of the compounds that need to be methylated to be used, and where this methylation process comes into play, phosphatidylcholine, which is needed for cell membranes, cell walls. It has to be methylated so that it can be used. Creatine is also produced with this, and melatonin. So serotonin gets methylated into melatonin so we can go to sleep. So different things that have to be methylated to be expelled by the body. Arsenic, histamine, and estrogen too. And so, histamine and estrogen do kind of go hand in hand. A lot of women will notice that when their estrogen levels go up, their histamine levels go up as well. So they'll be, have more of those types of symptoms with itching, getting hot, the heat, maybe a little rash kind of thing. So again, we see where all these things kind of coincide and kind of play on each other. So again, it's just kind of miraculous how complex our body is and how everything works together so well. So anyway, that's all for today. That's all for kind of a quick background on methylation, quick background on genetics and epigenetics. Next time, we'll get real specifically into MTHFR, probably a few other genes as well. And then just, talking about the different types of SNPs that can occur with MTHFR and how that may possibly play out. And then we'll share some different, resources too, where you can learn more about it. Because there are definitely some people where, this is kind of what they've studied for a long period of time. And that way, once we get into that, I think we can get into the question about different supplements and why that may work and why that may not. Again, it's going to kind of be a little bit of an experimental process. But I appreciate you guys watching. Thank you so much. Let me know if you have any questions. Hopefully I explained this well. Like I said, try to keep it somewhat basic and easy to follow along. But wanted to give some of those cofactors so you see why it's important to eat a healthy diet, to try to get different things in, and why supplements can be helpful in different cases too. But again, I just appreciate you guys so much. Please like, subscribe, do all that fun stuff, and I will see you guys next week. Bye.